So straightforward, guys. We'll be talking about the roles of cyclins and cyclin-dependent kinases in the regulation of the cell cycle, right? At least everybody here should know what the cell cycle is, right? That's a G0 to G1, okay, to the S phase, to the S, to the G2, to the M phase, okay? So basically, we discussed cell cycle at the elementary levels before, where we just spoke about the stages, okay, but we didn't actually talk about the regulation, right? So coming to the higher levels now, we're trying to see that this cell cycle is regulated by cyclins and cyclin-dependent kinases, right? And these two molecules are basically the molecules I will be talking about, how they are able to regulate the cell cycle and at what stages they are able to regulate the cell cycle. So let's start the story now. We said our cell cycle is divided into four phases. That's G1, S, um, G1, S, the G2, M. Okay, with a quiescent G0 phase. All right, the G0 phase, you see that the G0 phase is kind of like outside. Some people don't really um, include it as phase, okay, of cell cycle, but it's kind of like a phase though. Okay, so guys, the orderly progression of cells through the various phases of cell cycle is orchestrated or is coordinated by cyclins, cyclin-dependent kinases, and cyclin dependent kinase inhibitors. Do you understand? So now these cyclins are synthesized during specific phases to form complexes with and activate what? Cyclin dependent kinases, because these kinases are cyclin dependent, all right? So cyclins need to be secreted or synthesized before the cyclin dependent kinases are formed, all right? So now the cyclins are synthesized during specific phases to form complexes with and activate what? Cyclin dependent kinases, which are expressed constitutively throughout the cycle. So the cyclin now, earlier on the cyclin activated the cyclin dependent kinases and it will still bind with the cyclin dependent kinase, all right? So now when cyclin bind with cyclin dependent kinases, form what? A cyclin dependent kinase complex. Okay, now this cyclin dependent kinase, which is basically the complex that what drive the cell cycle by phosphorylating critical targets through within the cell. Okay, as the cell cycle is going on, this cyclin dependent kinase is the one that is basically trying to phosphorylate the whole cycle. So on the basis of this, we say that we have more than 15 cyclins, but the commonest ones are the what? DEAB. Okay, that's for cyclins. Then for the cyclin dependent kinases, the most commonest one are the what? 6, 4, 2, 1. All right. The mnemonics I actually use for this is decreasing in 2, then 1, right? 6 minus 2, 4. 4 minus 2, 2. Okay, then 1, right? Then this one I just call it deep. Do you understand? So now, you said that cyclins will bind to cyclin dependent kinases, all right? So if there are different cyclins and there are different cyclin dependent kinases, then they are basically binding at different levels, okay? So the commonest complexes are cyclin D will bind towards cyclin dependent kinase 4, cyclin E will bind towards cyclin dependent kinase 2, cyclin A will bind towards cyclin dependent kinase 2, cyclin B will bind towards cyclin dependent kinase 1, okay? So now, these complexes are inhibited by what? Cyclin dependent kinase inhibitors, okay? That's a CIP stroke KIP and the INK4 stroke ARF families, right? So the full meanings mean CIP is cyclin dependent kinase interacting protein, KIP is a kinase inhibitory protein, then the INK4 is the inhibitors of what? Cyclin dependent kinase 4. Okay, so this is basically, you could say this, how the cyclins are basically forming complexes with the what? Cyclin dependent kinases. All right, So on the basis of this, we say that the CIP and the KIP family comprise of what? P21CP, P27KIP, and P57 what? KIP2. Okay? The INK4 and the AR4 family comprise of what? P16 Inc 4A, P17 Inc 4B, P18 Inc 4C, and the P19 ARF. Okay, these are basically the cyclin-dependent kinase inhibitors. Now, the P16 Inc 4A in particular will mainly inhibit the cyclin D and cyclin-dependent 4 complex, right? While the P19 ARF helps to inhibit the degradation of what? P53 by what? Mouse double meanings to homolog, right? And this gene that is found in human beings too. Now, the internal control of cell cycle takes place at 2 checkpoints, right? And 1 restriction point. Okay. Now, the first checkpoint is what? Checkpoint that is in between uh, G1 and the S. Okay, the G1 and the S, you have a checkpoint there, and this checkpoint checks for DNA damage before the S phase. Okay, while the second point is what? Between the G2 and the M. Okay, and this will check for DNA damage before M phase. Do you understand? So GS, G1S to check for DNA damage before S phase. So G2 and M to check for DNA damage before the M phase. So now, though the S phase of the cell cycle is regarded as a point of no return where the DNA replication takes place, but any DNA damage after replication can still be repaired at the second checkpoint, since the separation into sister chromatids take place only at the M phase, right? So this is just a picture summarizing the roles of cyclin and cyclin-dependent kinases in the regulation of cell cycle. Okay, so you could just pause this video and get to like write this thing down and watch this video again to actually relate what is here to what we have there. Okay, so moving forward, we said that these 2 checkpoints have sensors and transducers. The sensors are made up of rad proteins and ATN, that's the ataxia telangiectasia mutated genes families, while the transducers include the CHK kinase family, and that's a checkpoint kinase. Okay, so following DNA damage from ultraviolet light, the P53 gene arrests the cell cycle through transcription of P21 and GAD45 that now repairs the damage. Okay, but if not possible, the P53 activates pro-apoptotic genes, and these are the genes that will basically what induce apoptosis, right? And we have the BAC, we have the BAD, we have the BAX, we have the BEAD, and we have the BEAM genes, right? So now, while the P53 gene acts mainly on the first checkpoint, the ATM gene acts on both points by sensing what double-stranded DNA breaks, arresting the cycle through inactivation of CDC25 phosphatase. That's the cyclin-dependent complex, right? So the RB gene also acts at the restriction point, that's the tail end of the G1, where the active RB, that's the hypophosphorylated, is bound to the E2F, thus preventing cell proliferation. Okay, so guys, this is basically the roles of cyclins and cyclin-dependent kinases in the regulation of the beautiful cycle of cell. All right, so that's it, guys, and see you guys.